So we are now discussing the hypothalamus pituitary gonadal axis, and we will discuss first the male reproductive neuroendocrinology. Of course, these are the organs that we're familiar with: the pituitary gland, the anterior pituitary gland. The hypothalamus is an glandular tissue, and gland 3 in the male, the testes, as the target organs. So the hormones basically are going to be the same, hormones 1 and hormones 2, the GnRH, the LH, and FSH. So that's gonadotropin-releasing hormone and luteinizing hormone, and follicle-stimulating hormone. And the target cells are in the testes, and we'll review the anatomy of the testes to look at these target cells, the sustentacular cells, and the interstitial cells. Anybody tell me another name for the sustentacular cells? The eponym. Sertoli cells. So the Sertoli cells are the eponym. It's named after whoever discovered it, Dr. Sertoli, I guess. And what about the interstitial cells? Leydig or Leydig. So those are the eponyms, and these are the nomina anatomica terms for those structures. So we stick with those nomina anatomica terms as much as we can. And then, of course, we'll discuss feedback regulation of the hormone secretion and other effects of the testosterone hormone tree. So again, gland 1, gland 2, gland 3, hormone 1, hormone 2, hormone 3, or hormones. And these are small neurons, so the neurons that are controlling the anterior pituitary have a short distance projection. So it's not that much shorter than the posterior pituitary, but their task is to produce a concentration in the plasma of the portal circulation sufficient enough to target the anterior pituitary cells. So they are smaller in their machinery. They're located in the preoptic area and the arcuate hypothalamic nuclei. And there's a lot known about these two populations of neurons as far as producing the tonic levels of the gonadotropins, and in females, a pulsatile change to trigger ovulation. In males, they both basically operate for the tonic and, of course, are influenced for circadian rhythmicity of the gonadal steroids. At what point in the day in the circadian rhythm are the male gonadal steroid plasma levels the highest? In the early morning. So gonadotropin-releasing hormone is synthesized and secreted by those parvocellular neurons into capillary bed 1. Target cells in the adenohypophysis are the gonadotropes. These gonadotropes, although there's a variety of their population and a lot of detail known about them, basically we can generalize and say that each gonadotrope expresses both the LH and FSH gene or genes. Is there more detail that needs to be added to that? Is there one gene for LH and one gene for FSH? These are heterodimeric proteins. Okay, so the alpha subunit and the beta subunit. The alpha subunit is expressed in gonadotropes and thyrotropes. So when I say the LH and FSH genes, I'm referring specifically to the beta subunit of those. So those are the hormones 2, the gonadotropins. And the gonads, of course, are the target, the testes and the ovary. And we'll review the details first for male and then female today. The gonadal steroids, testosterone, estrogens, and progestins. So in the male gland 3, testosterone is the major hormone. Of course, androgens are the collective term for the male steroids, and testosterone is only one of those. And I think we have a slide that illustrates a few others. There are various stimuli, circadian, autoregulated for the menstrual cycle, and regulation is the LH and FSH are going to, through the short loop negative feedback, inhibit GnRH synthesis and release, and then through the long loop negative feedback, the gonadal steroids will inhibit at both the level of the pituitary and the hypothalamus, but there's debate in the literature as to whether that's a direct or an indirect negative feedback on the GnRH neurons, because for a long time, the methods were not sensitive enough to detect expression of the gonadal steroid receptors in the GnRH neurons. So there was no receptor, there couldn't be a direct negative feedback. So that's still under intense investigation. So again, from the paraventricular nucleus of the hypothalamus to the capillary bed 1, through the portal vessels to capillary bed 2 for GnRH, targeting the gonadotropes for the synthesis and secretion of FSH and LH, and ultimately targeting different target cells within the target glands, the gonads. So again, we're back here in the anterior pituitary inside that capillary following the GnRH molecule. We're sitting on it. Diffuses down its concentration gradient through a fenestra of the endothelium of this capillary, discontinuous capillary, because it has the discontinuous cytoplasm. So we float right through that window. We're bouncing around in here. In this field of view, we'd be lost because we only have growth hormone or somatotropes and prolactin hormone synthesizing and secreting neurons, the lactotropes. So we'd bounce around and we wouldn't do much good unless we could diffuse far enough away and find a gonadotrope, to bump into and bind to our receptor. Again, putting ourselves in those subcompartments of living organisms is extremely important, because if we're thinking of hormones and drugs and pathogens, those are where they are, and we have to keep that in mind all the time. What is the appearance and the structure and the boundaries, and what are the chemical properties of those areas? What type of connective tissue, what type of proteins, what type of immune cells, and a variety of things like that. So that micro-scale anatomy is obviously one of my favorites, especially putting it within a functional context. So in the testes, then, looking at target gland for target glandular tissue, because it produces gametes as well as the hormones. There are the 3 cell types in the male target gland tissue, the testes: the sperm-producing cells, the Sertoli cells, and the interstitial cells. So are the sperm-producing cells direct targets for gonadotropins? Are they direct targets for hormone 3? Testosterone will impact the sperm-producing cells, and we'll see that there's a differential targeting of the, anybody now remember the nomina anatomica for the Sertoli cells? They sustain development of the sperm-producing cells, so it's the sustentacular cells. So this is a cutaway view of the top portion of a testes, and we see all the connective tissue septae separating it into these compartments, and of course each contains a long coiled tubular structure, the seminiferous tubules. We take a closer look at a histological section, and so we're cutting through in cross-section 2 adjacent and a third that is partially within our field of view, seminiferous tubules. So we have to think again of subcompartmentalization inside the lumen of the seminiferous tubule, the epithelial lining of the seminiferous tubule. So this portion close to the lumen is adluminal, AD. So we use adhesive to get things to stick together. When you are abducted, you're taken away. So here's the abluminal, AB-luminal portion of the epithelial layer of the seminiferous tubule. Outside of and among, we have 3 here, so we have to use among the seminiferous tubules are the interstitial cells of Leydig. So we note also there are capillaries, blood vessels are in here. I don't see a capillary right in here, but very likely there was one before the histological processing. Endocrine tissue is highly vascularized. So we are now following on top of LH and FSH, diffusing out into the interstitial fluid, and we are either going to bounce into the interstitial cells or have to get through the connective tissue capsule of the seminiferous tubules and bind to the sustentacular cells. So let's take a look at the specificity of the targeting of FSH and LH. So the LH is targeting the Leydig cells, so fortunately it's L and L, and these interstitial cells are the source of the testosterone, as we had said. So the secondary messenger system we'll discuss in a while. It's, of course, the adenylate cyclase producing cyclic AMP, and it impacts the biosynthetic enzymes. In the case of lipid synthesis, we always have to think of the intracellular signaling mechanisms for these metabotropic receptors as targeting the enzymes and the genes for those enzymes. So if we activate the gene for the rate-limiting enzyme for this pathway, biosynthetic pathway, and we also activate some mechanism, a kinase, a protein kinase, which would be protein kinase A for the cyclic AMP system, and we phosphorylate that rate-limiting enzyme and increase its activity, then we have increased synthesis of the steroid. When a steroid is synthesized, is it packaged and stored? No, you can't. How do you create a localized concentration of a lipid-soluble molecule? Got to have something that'll bind it and hold it there. So let's take a look at what happens when the FSH targets the sustentacular or Sertoli cell. Same second messenger system, except now we're targeting a gene for androgen-binding protein, and the androgen-binding protein will be localized in that cell, and it will help to hold testosterone nearby so it can bind to its androgen receptor and have impacts on the Sertoli cell, which will support or sustain the development of the sperm. As well, of course, we need to back up. The testosterone will also diffuse away in the plasma of those capillaries, venous return through the heart and lungs and everything, and systemic delivery to the target organs for the secondary effects at the body. So here we see a question, which is at a rather high level, outside the realm of this course, but we still should be able to think about it, and we'll see gene structures and the regulation of genes in a moment. So the LH receptor is going to act through the second messenger system, increasing intracellular cyclic AMP. Cyclic AMP phosphorylates, well, that should be pKa to go along with that. Sorry about that. Oh, so bad. pKa for the A in cyclic AMP. And the CREB, which is cyclic AMP response element binding protein, and the CRE is cyclic AMP response element on the target gene. So we'll put this together, and while we're introducing these terms, and you're going, what in the heck is he talking about? But don't worry about it. We'll piece it together. We all know what a gene looks like. We should understand promoter regions. That's where we get tissue-specific and cell-specific gene regulation, expression regulation. And as well, we have other factors, proteins that can bind there, like the steroid hormone receptors, that can target gene expression, increasing or decreasing the expression of a gene within a cell. There are other spans of DNA on that promoter region upstream of where we start transcription that also can bind proteins, and any protein that binds to DNA and changes the expression of a gene is called a transcription factor. So in endocrinology, you have to have some background knowledge about transcription factors and how they work. And we'll build on that. We're just introducing what happens, what the LH is related to, and now here the FSH receptor, also coupled to the cyclic AMP second messenger system. So the FSH is binding down here to the abluminal membrane, or basal membrane, because this would be the apex of the cell and the base. The abluminal or basal membrane of the sustentacular cells, activated adenylate cyclase, picture cyclic AMP levels building up. I got to move the pip here. Picture the cyclic AMP levels building up in here, activating pKa. I've got C here again. and that'll phosphorylate CREB. CREB will get into the nucleus and bind to the CRE on a gene, say the androgen binding protein gene, and increase the expression of that gene. Very likely, the pKa will also phosphorylate other intracellular signaling mechanisms to alter the general metabolism of this cell, such that it sustains the meiotic production of the male gamete, the sperm. So let's start at the membrane and work our way in. Water-soluble molecules act at the level of the membrane. So we are focusing on the left-hand side, where we have an activation of the effector, the adenylate cyclase. Other hormones might impact it to inhibit it. So this would be a G sub S linked protein, G sub S, the S for stimulate. So the FSH that we'll focus on for now, and of course LH is acting through this as well, in addition to several other hormones, of course. And the FSH then is binding to the receptor, and that will alter the G protein so that it's activated. It'll lose the GDP and bind GTP, and then become dislodged from the receptor protein, binding protein, and it can move through the membrane. This is the fluid mosaic model, and when it contacts the adenylate cyclase, it activates it, and nearby ATP, of course, will be enzymatically converted to cyclic AMP, which will target, it'll diffuse away and target inactive protein kinase and cause a change of that, a catalytic change, and release the active protein kinase that triggers several other intracellular mechanisms. It can activate enzymes and stimulate cellular secretion and open ion channels and a variety of other things. So let's see what might happen at a gene when we have regulated transcription by the pKa phosphorylating CREB, cyclic AMP response element binding protein. It is a transcription factor. So the CREB is in the nucleus. It gets phosphorylated by the pKa and now bound to the cyclic AMP response element or regulatory element on the promoter region upstream of the start site for RNA transcriptase binding. And it alters the environment in this area, and it increases the probability that RNA transcriptase will bind to there's the TATA box and all that stuff here for the actual binding of the RNA transcriptase. And it will either enhance or inhibit, so it either increases the probability or decreases the probability. It can be either direction depending upon the cell and the gene. In our case, we're picturing this as FSH causing this, and this being the androgen binding protein. We know every gene has exons and introns. This one is illustrating 3. It might be more than that. So this is how we impact synthesis and the other mechanisms for activating proteins and ion channels in the membrane are these release mechanisms. So in the case of the interstitial cell, LH binds to its receptor, and it's going to activate through the cyclic AMP the pKa. The pKa is going to activate a variety of enzymes along the way from the cholesterol a little bit farther back through pregnenolone and then finally to testosterone. So it's going to phosphorylate those enzymes and also increase expression of their genes, because as we phosphorylate enzymes, we recruit those enzymes from an inactive pool to an active pool, and then they're on their way to being inactivated again and very likely degraded and recycled. So at the same time that we activate and pull out of a reserve pool, same thing for the release, we have to turn on the gene to synthesize to replace what we just consumed. So always keep that in mind. If you use something, you got to replace it. Okay, so if you share a refrigerator where you live, you drank the last beer. Well, the Spurs, they won last night, right? So you drank that last beer, you got to replace it. So it's the same type of phenomenon. So we behave like ourselves. So the testosterone can also be altered enzymatically. And this step here is very important in neurons and actually at the anterior pituitary. In males, we express the gene in those cells, those particular target cells, for this enzyme aromatase that aromatizes this ring A here. You recognize this, isn't that the phenol thing with all those double bonds in there? So it's an aromatic ring now, and it has two hydroxyl groups on it and a methyl group, and that is estrogen. And then in those cells are males' anterior pituitary gonadotropes and hypothalamic neurons and brain neurons that are targeted by testosterone in many of the areas. The testosterone is converted to estrogen, and those cells express the estrogen receptor gene, and the estrogen binds to the receptor in those cells and produces what we see testosterone doing in males, impacting some behaviors. Of course, testosterone has its own androgen receptor in many cells as well, and that has the impact on, of course, sexual drive and aggression, and as well that capability exists in females. In females, testosterone is the major hormone for stimulating sexual receptivity, as it is in males. And what would be the source of testosterone in females? Adrenal cortex, the zona reticulata, but as well, the ovaries produce testosterone on the way to producing estrogen, and some of that testosterone diffuses away. So we all remember our biochemistry. You start with enzyme 1 and enzyme 15 or whatever, and you produce those short-lived intermediaries. But depending upon the enzyme somewhere along the way that finally makes converts the aromatase, if we're producing more testosterone than the aromatase can draw to the estrogen, then some of that stays around for a period of time and can diffuse away. So all that biochemistry is important, very important, understanding how we synthesize a multitude of steroid hormones, and not just the final hormone is present in the plasma and in the interstitial fluid compartments. Some of the intermediaries are produced at levels and diffuse away from the enzyme next in the chain before it can act on it. Testosterone in some target cells is also acted on by 5-alpha reductase to produce 5-alpha dihydrotestosterone, and that will target the androgen receptor. So testosterone isn't the final hormone in communication. It actually is either 5-alpha or the estrogen. So here we see two boxes, one showing the Leydig cell and one showing the Sertoli cell. So the LH binds to its receptor in the Leydig cell, activates adenylate cyclase, increases cyclic AMP, and look at it, it's targeting a hydrolase that removes the cholesterol from its ester bond. It's the only way you can hold cholesterol in one place, cause an ester bond and covalently stick it someplace. Right? So the only way you cause a localized concentration of a steroid is you got to hold it there one way or another, either through dynamic ionic bonding, all those van der Waals forces and stuff that you recall from chemistry, or covalently bond it there and hold it securely. In the female, the corpus luteum is the yellow body because so much cholesterol ester is stored there that it actually looks yellow in fresh tissue. So a hydrolase is activated to break that down, and then we get cholesterol, and then the mitochondrial transport side chain cleavage enzymes are activated by the cyclic AMP. And of course, this isn't an on-off. There's just a population of these enzymes, and some of it is active, and some of it's being inactivated. Now the cyclic AMP comes in, and it contributes more to that active pool to be converting from the cholesterol to the pregnenolone, and then hydroxylase is also targeted. So we pump up these early enzymes, and we just drive it through the system. Here, take note of the dehydroepiandrosterone. Anybody know the acronym for this, the letters for that? DHEA. You might see that on the shelves at HEB or other stores. It's a common over-the-counter compound with a variety of claimed things. In the lecture that I'll be posting on the stress system, I take you off to a website to show you an application for use in dogs to control the body weight of dogs. The androstenedione is then produced, and then the testosterone, and then the testosterone diffuses to the Sertoli cells, the sustentacular cells, and here the FSH is binding, increasing cyclic AMP that activates the aromatase. Now, every time we see an arrow going to one of these enzymes to activate it, we've consumed an enzyme, basically, and there's a parallel pathway to the nucleus to activate the gene or increase the expression of the gene for that enzyme, because we've got to kick back into the inactive pool, our reserve, to feed into this every time we activate an aromatase. So in the Sertoli cell, actually the testosterone is converted to estradiol, and it binds to its receptor and has the effect. So I know it's a lot of detail. There's no harm in seeing it. There's no harm in struggling to remember it. I'm not going to be gauging your performance in this class. In other words, it won't impact your grade if you can't work out all those details. There's no harm in seeing it happen. Biology is, of course, splendorous, and it's awing us all the time. So exactly how does the feedback work for the steroid hormones? So here we see hormone 1, hormones 2 targeting their particular cells, and a couple of hormones being produced. Inhibin also is a peptide hormone also produced by the Sertoli cells, and it has a direct negative feedback specifically on FSH. The testosterone has its negative feedback, as we had discussed. So now we're back here. The inhibin and the testosterone are in the plasma of this capillary of the anterior pituitary. Diffuses down its concentration gradient, doesn't need the windows. There's the gonadal steroid or androgen binding proteins, carrying proteins are present in the plasma, so they hold it in the plasma. Plasma concentration can be a bit higher, diffuses out into the cytoplasm, into the nucleus, binds to its receptor, and then now the ligated receptor can bind to the either the androgen response element or the estrogen response element on the different target genes. So lots of genes in here. Which ones can be targeted? Only those that have that little stretch of DNA in their promoter region that can bind the ligated receptor. So we saw Krebs was a transcription factor. It has to be phosphorylated to bind to its strand of DNA on the promoter region, or its RE site. The steroid receptors are also transcription factors, but they're ligand dependent. They have to be ligated by their cognate hormone, and then there's a conformational change. Then it can bind to the response element. So let's take a quick peek at that. Up here in the top right, the steroid is coming through the cytoplasm into the nucleus. Here we see the receptor that's complexed with some chaperone proteins. And when the hormone is ligated, changes conformation, has a lower affinity for the chaperone proteins. They fall off. It now actually exposes its DNA binding domain. So it has a higher affinity to bind to the DNA. And there is a DNA-associated receptor there, or in our case, we're talking about testosterone being converted to androgen, so it's the ARE, the androgen response element, or if it's converted to estrogen first in the cell and then binds to the estrogen receptor, would be an ERE, the estrogen response element. And we change transcription. They always take this to new protein, but if we're talking about negative feedback, it's a decrease in the transcription of this, a decrease in the messenger RNA in the nucleus, a decrease of translation rate in the cytoplasm, and a decrease of the protein. So keep that in mind. It can go in either direction. So now we look at this same model gene. Nothing changes over here. We have just a different response or regulatory element. In this case, it's for the estrogen receptor that is ligated, so it now has a high affinity for the DNA, the estrogen response element. When it binds, it will change the expression of that gene, again, by altering the probability that RNA transcriptase will bind appropriately and transcribe this gene. So we ask the question, what genes might be targeted? Well, in the anterior pituitary, we immediately know it's the beta gene for FSH and LH, right? In the hypothalamus, at present, although they have found the ERE, they haven't found the estrogen receptor in the GnRH neurons. GnRH gene has the ERE, so there's still this thing that we don't know. Reproduction is still an enigma. So the ER is a ligated estrogen receptor protein. It is a transcription factor like many others. So here is a model structure of the intracellular receptors for the steroid hormones. There's a large family of these, which includes for the thyroid hormone and vitamin D, as well as the steroid hormones. So there's the amino terminal and the carboxy terminal, and 3 domains. There's the hormone binding domain. When it's ligated, it changes conformation and exposes the DNA binding domain. And then the amino terminal end of this interacts with other transcription factors. So it brings in other proteins, either to clutter it up so the transcriptase can't get there, or to create maybe more uncoiling of the DNA so that the more transcriptase can randomly bounce into and bind. So the negative feedback on the FSH genes would decrease that gene expression.